Regulation of glomerular filtration. The glomerular filtration rate, or GFR, must be precisely controlled. If it's too high, fluid flows through the renal tubules too rapidly for them to reabsorb the usual amount of water and solutes. Urine output rises and creates a threat of dehydration and electrolyte depletion. If the glomerular filtration rate is too low, fluid flows sluggishly through the tubules. They reabsorb wastes that should be eliminated in the urine, and a condition known as azotemia may occur. The only way to adjust this glomerular filtration rate from moment to moment is to change the glomerular blood pressure. This is achieved by three homeostatic mechanisms: renal autoregulation, sympathetic control, and hormonal control. Renal autoregulation. Renal autoregulation is the ability of the nephrons to adjust their own blood flow and glomerular filtration rate without external control. It enables them to maintain a relatively stable glomerular filtration rate, in spite of changes in arterial blood pressure. If the mean arterial pressure rose from 100 to 125 millimeters of mercury and there were no renal autoregulation, urine output would increase from the normal 1 to 2 liters per day to more than 45 liters per day. But because of renal autoregulation, urine output increases only a few percent, even if the mean arterial pressure rises as high as 160 millimeters of mercury. Renal autoregulation helps to ensure stable fluid and electrolyte balance in spite of the many circumstances that substantially alter one's blood pressure. There are two mechanisms of autoregulation: the myogenic mechanism and tubuloglomerular feedback. The myogenic mechanism of renal autoregulation. This mechanism of stabilizing the glomerular filtration rate is based on the tendency of smooth muscle to contract when stretched. When arterial blood pressure rises, it stretches the afferent arteriole. The arteriole constricts and prevents blood flow into the glomerulus from changing very much. Conversely, when blood pressure falls, the afferent arteriole relaxes and allows blood to flow more easily into the glomerulus. Either way, glomerular blood flow and filtration remain fairly stable. The tubuloglomerular feedback of renal autoregulation. This is a mechanism by which the glomerulus receives feedback on the status of the downstream tubular fluid and adjusts filtration to regulate its composition, stabilize nephron performance, and compensate for fluctuations in blood pressure. It involves a structure called the Juxtaglomerular Apparatus. The Juxtaglomerular Apparatus is found at the very end of the nephron loop, where it has just re-entered the renal cortex. Here, this loop contacts the afferent and efferent arterioles at the vascular pole of the renal corpuscle. Tubuloglomerular feedback begins with the macula densa, which is a patch of slender, closely spaced sensory cells on one side of the loop. When the glomerular filtration rate is very high, the sodium chloride level in the nephron loop is also sharply elevated. Cells of the macula densa absorb sodium, potassium, and chloride ions, and water flows osmotically. The cells swell and secrete ATP from their basal surfaces. The ATP is metabolized to adenosine by nearby mesangial cells. Adenosine acts as a paracrine messenger that stimulates the nearby juxtaglomerular cells, or simply granular cells. These are modified smooth muscle cells wrapped around the afferent arteriole and, to a lesser extent, the efferent arteriole. They respond to a rising adenosine level by constricting the afferent arteriole. This reduces blood flow into the glomerulus, thereby reducing the glomerular filtration rate and completing a negative feedback loop. The mesangial cells of the glomerulus may also contract, constricting the glomerular capillaries and reducing filtration. Mesangial cells also form a supportive matrix for the glomerulus and phagocytize tissue debris, preventing the glomerular filtration membrane from clogging up. Just beyond the juxtaglomerular apparatus, the distal convoluted tubule has a limited capacity for sodium chloride reabsorption. The tubuloglomerular feedback process may help to prevent overloading the distal convoluted tubule with sodium chloride, and thus prevents excessive salt loss and water loss in the urine. Granular cells are named for the fact that they contain granules of renin. Renin is secreted from the granular cells in response to a drop in blood pressure. This initiates a renin-angiotensin aldosterone mechanism, which restores blood pressure and supports blood volume. Two important points must be noted about renal autoregulation. First, it doesn't completely prevent changes in the glomerular filtration rate. Like any other homeostatic mechanism, it maintains a dynamic equilibrium. The glomerular filtration rate fluctuates within narrow limits. Changes in blood pressure do affect the glomerular filtration rate and urine output. Second, renal autoregulation cannot compensate for extreme blood pressure variations. The mean arterial pressure, or MAP, range of 90 to 100 millimeters of mercury, the glomerular filtration rate remains quite stable. Below 70 millimeters of mercury, however, the glomerular filtration and urine output cease. This can happen in what's called hypovolemic shock. Sympathetic nerve fibers richly innervate the renal blood vessels. In strenuous exercise or acute conditions such as circulatory shock, sympathetic stimulation, and adrenal epinephrine constrict the afferent arterioles. This reduces the glomerular filtration rate and urine output while redirecting blood from the kidneys to the heart, brain, and skeletal muscles, where it is more urgently needed. Under such conditions, the glomerular filtration rate may be as low as a few milliliters per minute. The renin-angiotensin aldosterone mechanism. Any substantial drop in blood pressure due to bleeding, dehydration, or other cause is detected by the baroreceptors in the aorta and carotid arteries. They transmit a signal to the brainstem, leading to various corrective sympathetic reflexes. One such response is that sympathetic fibers stimulate the granular cells to secrete the enzyme renin. Renin acts on angiotensinogen, which is a protein in the blood plasma, to split off a 10 amino acid peptide called angiotensin 1. In the lungs and kidneys, angiotensin converting enzyme, or ACE, removes 2 more amino acids, converting it to angiotensin 2. Angiotensin 2 is a hormone that acts in several ways to restore fluid volume and blood pressure. Angiotensin 2 is a potent vasoconstrictor. Widespread vasoconstriction raises the mean arterial blood pressure throughout the body. In the kidneys, angiotensin 2 constricts the efferent arterioles and, to a somewhat lesser degree, the afferent arterioles. By constricting the glomerular outlet more than the inlet, it raises the glomerular blood pressure and glomerular filtration rate, or at least prevents a drastic reduction in the glomerular filtration rate, thus ensuring continued filtration of wastes from the blood even when the blood pressure falls. Constriction of the efferent arteriole lowers the blood pressure in the peritubular capillaries downstream. Since capillary blood pressure normally opposes fluid reabsorption, this reduction in blood pressure strongly enhances the reabsorption of sodium chloride and water from the nephron. More water is then returned to the bloodstream instead of being lost in urine. Angiotensin 2 stimulates the adrenal cortex to secrete aldosterone. Aldosterone promotes sodium and water reabsorption by the distal convoluted tubule and collecting duct. Angiotensin 2 also directly stimulates sodium and water reabsorption in the proximal convoluted tubule. It stimulates the posterior pituitary gland to secrete antidiuretic hormone, which promotes water reabsorption by the collecting duct. Angiotensin 2 stimulates a sense of thirst and encourages water intake. The renin-angiotensin aldosterone mechanism collectively raise the blood pressure by reducing water loss, encouraging water intake, and constricting blood vessels. Thank you for watching.